everybody for coming. I appreciate everybody who came, and Mom, thanks for coming, and Ashley and your family, thanks for coming, and my girls. So anyway, I really appreciate everybody being here, and so just Dick, thanks for coming all the way from Michigan. It's nice. So I'll just jump right into it because we have a lot to get through. So this is going to be kind of a condensed version of everything that I did during my time here. Thanks. Yeah. So basically, I'm going to be talking to you about one of the big components of my research, which is looking at, like Simon was saying, this big family of genes called the Calmodulins and Calmodulin-like proteins, and a few of those that are especially interesting to us. So this is the poster that Sarah Friedrich helped me put together for the defense, and I chose it. If it's not obvious now, but what we're talking about is kind of the interaction between mechanical stimuli. So in this case, a person looks like from the 40s sneezing on a plant, and this plant is Arabidopsis. That's our lab rat plant that we all plant molecular biologists work on. And mechanical stimulus is the sneeze hitting the plant, and the plant will have a particular reaction to that. And then unavoidably, there will likely be a pathogen stimulus in there in the form of bacteria or viruses or mold spores, that sort of thing. And so what I'm going to talk about is kind of how plants react to each of those things and how there appear to be some shared pathways involved there, some overlapping, maybe some crosstalk. So what brings these two things together is something that's called calcium signaling, and I'm going to tell you a little bit about that. So calcium, what is it? Well, we all are familiar with what doctors recommend to humans is that we have a certain amount of calcium in our diet so that we have strong bones and strong muscles, and also making sure that our nerves fire when they receive a stimulus. You have an influx of calcium at the axon terminus, and then that allows neurotransmitters to be emitted. And so plants kind of do similar things. They signal with calcium. Without calcium, we wouldn't survive very well. Like I said, calcium is a signaling molecule. We regard it as a second messenger, which means that when there's a stimulus, so if there's a bacterium or some sort of stretching of the cell, that sort of thing, that's the first message that the cell gets, that initial stimulus, and then calcium acts as a second messenger. So an additional way of telling the cells that there's something going on there. So that's what calcium is. You're going to see calcium here, this green orb, show up in quite a few places, and because it's important for what I'm going to be talking about. An important thing to point out is that maybe you didn't know, but plants can't run away from their pursuers. They're kind of stuck where they are, and so they need a way to react to their environment. And for instance, if there's microbes or insects or wind and rain and that sort of thing, and adapt to what's going on and strengthen themselves. And then in order to do that, one of the first things, like I said, this is the signaling molecule calcium. It indicates that there's that stress there. So they flood their cells with calcium, and that communicates what's going on. And then they need a way to interpret that signal, and so that's one of the big things is we have proteins that reside inside the cell that take this input of calcium, an influx of calcium into the cell. They perceive that influx and then go and do something with it. And the family of genes and proteins that I'm going to be talking about, and Simon mentioned, is the Calmodulins, and they are very important to this process. They bind calcium and do something with that information. And they also have related members of their family, which are called the calmodulin-like proteins. And that kind of expands this suite of proteins that can be responsive to calcium. And then they go on to regulate numerous targets. Finally, it's really important to have this ability to bind the calcium and do something with it, because if you don't, and there's a pest or something like that, and plants don't respond, then it can have some really, really negative effects. And the calcium, loss of this calcium binding can be really, really detrimental to the plant. So I talked about calmodulins. Why don't I show you what one looks like? This is what a calmodulin looks like. It's a group of helices and these calcium binding domains here. So notice on the typical calmodulin member, a family member, you'll have one calcium here, one here, and one here, back here, and then one back here as well. That's when it's bound to all 4 calcium ions. These are found in these loops, and I'll talk to you a little bit about those loops. So we made a review, we wrote a review, and for one of the figures, I recapitulated a figure from 1974, describing how calcium binding in these loops works. So you have here from the tip of the forefinger down to the base, you have the E helix. It's called the E helix because of where it resides in the protein. Between the E helix here and the next one, you have this loop where there are proteins that coordinate this calcium ion, and then the end of the loop you have the F helix. And so we call it an E-F hand because of how it looks. And it holds the calcium in place, and once the calmodulin is bound 4 calciums, in this case, calcium ions, then you have the ability for it to change its shape and go on to bind other things like other proteins and DNA, so it can regulate other processes in the cell. How that's done is, here's our big calcium again. It's in the center, and each one of these amino acids sticking out here corresponds to one of these coordinates here. And so water is one way the calcium is bound in the pocket, or in the EF loop, and then you have amino acids at these different coordinates that hold it in place. So that's kind of like the molecular aspect of what's going on there. And then I wanted to talk a little bit about, so once there's a calcium signal and you have all these proteins that are going to go and bind to that calcium, well, what does the signal tell you? Well, we have models that kind of describe a little bit of what's going on here. So in the instance of like you have a touch stimulus, so you just brush the plant, you have a really immediate influx of calcium, and it behaves kind of like a wave. You get this peak of influx of calcium in the cell quickly tries to shuttle it out, and this is over often in a matter of minutes. So you get a quick increase in calcium. During this time, proteins come along and bind to that calcium, and then you get a reduction. The cell shuttles the calcium back out using pumps, and then a lot of times you get a second kind of sustained but lower increase in calcium and this shape of this wave, we think, forms kind of the important basis of what the stimulus-specific signal is. So for touch, you have one maybe that looks like this, but for other stimuli, you'll have different looking ones. And then calmodulins are tuned to different signals that have different shapes, and so you have a different calmodulin responsible for responding to a different kind of stimulus-elicited calcium increase. In this case, we have CML24, which is one of mine, calmodulin-like 24. It's also called TOUCH2. And it goes on and affects the shape of the cell wall. It helps rigidify things so they're less touch sensitive down the road. Then you look at kind of how complex that could be. You can have, in response to a particular stimulus, you can have one that's just a large, sustained increase in calcium inside the cell, so that cells have opened channels that allow the calcium to move across or with its electrochemical gradient into the cell. We have the same signature up here, then we have one that's kind of peaks and valleys, rapid increase and decrease in calcium, and they can correspond to different types of stimuli. And then so we have a whole suite of proteins that are responsive to these different calcium signals. Now what's cool about the Arabidopsis genome is there are all these different stimuli out there, and all these different stimuli may have different calcium signatures, is what we call these different waveforms, that are responsible for signaling that these different stimuli are there. And what's cool about the Arabidopsis genome is that there are 7 Calmodulins and 50 CMLs, like Simon said, 57 members of the family that could be, in theory, tuned to different stimuli, tuned to different calcium signatures. And there's been research looking into specific ones in response to drought, in response to flooding, in response to high light, that sort of thing. And to touch, and importantly, to touch in pathogens. And so I'm going to show you kind of what that looks like. These are all of them. These are their amino acid sequences. We clipped off the N-terminal domain there just to show you the 4 EF hands that bind the calcium. And if you were to go and look through all of these and compare them even back to human calmodulin at the very top here, you'll see that especially in the EF loop where calcium is bound, they're incredibly homologous. So they're conserved. Even if there's a different amino acid there, it can carry out the same function as the one that it's replacing in the way that it coordinates calcium. And then with a few exceptions, of course, as there always are. In red here, you have those EF hands in all of these calmodulin-like proteins. You have the EF hands that, because of either mutations over evolutionary time or loss of different amino acids, you've lost the function of that EF hand and therefore potentially the calcium binding ability. And so, for instance, for CML1, there's only one functional EF hand, which you can imagine would change its ability to bind calcium or the amount of calcium it binds, and maybe what kind of signature it's tuned to. You have a few here that have 2 functional EF hands remaining, so that could also have repercussions for its calcium binding. You have a few here that are 3 EF hands, and then you have one, which is CML12. It's also called touch3 because of its canonical or its understood involvement in the touch response, that has 6 EF hands. And I didn't show all of them here, but that's just because the first 2 are, these 2 are duplicated. They're identical. So you have the first 2, and then the first 2 again, and then the last 2, so you have 6. And then all the rest of them have the normal 4 functional EF hands for binding calcium. Now, we don't know what all of these do, obviously, because we're still working, people are still actively investigating, but we have a pretty good idea of what a lot of them do. I'm not going to obviously get into that because it would take forever, but we can look at these calmodulin and calmodulin-like proteins, and you can just kind of see how they're grouped together based on their evolutionary history or how related they are to the group 1 calmodulins, which are Cam 1 through 7 up there. There are a couple in there that I'm really interested in, Cam 2 and Cam 7, and then distributed throughout. As the number increases, you get further away in terms of relatedness. So how divergent they are from calmodulin. Group 6 is repeated here because they named a lot of these calmodulins before they realized their historical connection that they had to each other. So we had, they ended up being split, but in all, they're grouped in 9 different groups. And they're all involved, or purportedly or presumably they all are involved in stress responses, many different stress responses. And so I'm going to talk to you a little bit about a couple of those stress responses that I was really interested in. We are, as a lab, really interested in. So we knew, especially from kind of experience working with plants in the field or in gardens and stuff like that, that when you touch plants, or when they're grown outside, they are more resistant to pathogens. When you grow a plant inside in a growth chamber or a greenhouse, they don't have all these stimuli if you just leave them alone. They grow fine usually, but if you were to infect them with a pathogen in exactly the same way you did it to a plant grown in the field, they just wouldn't respond as well. They wouldn't survive as well. It's just something that we've observed over time. And so we knew that, but we didn't really understand how. And so one of my goals was kind of to get at maybe some of the molecular background, some of the underlying behavioral stuff going on that can help explain why that might be. So we did know that calcium signaling from research that's been done all over, and calcium signaling and these signatures are involved in both of these types of responses. And so maybe there is some sort of either overlap between these two systems for responding to these stimuli. So maybe they share the same proteins, so the touch response proteins might be similar to, or there might be a few that are shared with the pathogen response proteins, or there's some sort of communication going on between those systems, or it's kind of like one large network of interactions, and it's more nebulous, cloud-like, that you just you can't clearly say one's a touch response, one's a pathogen response. So we wanted to look at that. Clearly there are too many CAMs and CMLs for one person to test. But like I said, people have been doing work on this for a long time. So we looked at the literature to see what's kind of out there. And we found that a really important paper is published in 2005 by the lab of Janet Brom down in Rice University, had looked at the expression of genes in Arabidopsis in response to mechanical stimuli. In this case, they bent leaves back and forth and then looked at what the expression patterns of genes were like. And they found that one of their Calmodulins and 10 CMLs specifically were always upregulated, always expressed more than background levels, so more than just kind of like the wild type response, more than when they were unstimulated in response to touch. So we had that information, but we also wanted to see if there was more out there that maybe we were missing. So we used bioinformatics tools. So there's software that's available to researchers and everybody where you can go and you can look, you can say, either you can start with a list of genes that you are interested in and see kind of what connects them, or you can start with a particular response you're interested in, or a particular pathway, some sort of interaction that you're looking for, and you can query that software and say, yeah, I'm looking for this, maybe I can form a hypothesis around something that I find there. And so we did that, and we came up with some pretty cool networks that I'll show you in 2 slides. So first, I'm going to say which of the CAMs and CMLs, since it seems important. So CAM2, which is also called Touch1, I mean, it makes sense that it would show up in the touch response. That's nice that happened. CML5, CML9, CML12, which I said before is also called Touch3, 16, 24, which is Touch2, 27, 40, 44, 46, and 49. So this is the list of genes that I was looking at, and we wanted to see, okay, we know that from research that's been done that they are somehow involved in the response to touch, maybe because they increase in expression after mechanical stimuli. Now, we wanted to see, using these bioinformatics tools, can you actually form a coherent, like an intelligible network of genes where you can see clear connections between these genes and other genes and see what those connections mean. And so we did that. We plugged this list of genes in there into a service called String, which is really nice. It allows you to look at a whole bunch of different types of interactions that are out there. Now, we screened out interactions that are just based on like co-expression or not co-expression, co-presence in the literature. So if a paper talks about one gene and it also talks about another gene that doesn't really inform us of anything, they just happen to be talking about them. But we were looking for specifically if they are experimentally determined interactions between these genes and the proteins that they encode. And then also, have they been previously annotated as part of a known pathway of interaction? So people have spent a lot of time taking suites of genes and proteins and saying, all right, we know how this pathway, this molecular network works. So if we're looking at the pathogen response, we know that this one's involved and this one's involved. So it also queries those things. And it forms a network like this. And one thing that will stand out right away is, like, I have my genes here highlighted in red, all but one of them, so all but CML46, which is kind of up here in this cloud of genes, all of them but CML46 are connected to this hub here, which is NOA1, which is a known pathogen responsive. It's known to be somehow involved in the pathogen response. And so that gave us a good indication that maybe, maybe there's something to this idea that the touch response pathway and pathogen response pathway are there. But, we didn't know. We wanted to make sure that, there are probably all sorts of other types of connections going on up here that are bringing different genes into this putative touch responsive network. And so we made, over here, we made a separate network because you always have to have two of everything. So we took the same list of genes and said, okay, we just want connections that are related to, for sure, the pathogen response. And so what you have here are, this is from KNetMiner, which mines OnDex, which is ontology index, which means these genes have been, and their interactions have been annotated to be involved in such and such pathway. And so the ontology that we're talking about here is pathogen responsive or defense response, or FLAG22, which is an important molecule that I'm going to talk about in a few minutes. And indeed, once again, we took our list of genes and it did form a coherent network. They are connected and they're pretty closely connected to each other. And the large ones are my genes, just for emphasis, and then the little ones are some of the other genes that connect them. One thing you'll notice is that up here, there's touch4. That is not a calmodulin or calmodulin-like protein, but it is one of the well-studied touch response genes, so that's there. And then secondly, we have this guy, CAM7, who CAM7 is not one of the known touch responsive genes, but it is known to be pathogen responsive. And it's related to CAM2 over here. It only has a single amino acid difference. So there's just one tiny change, and you get, instead of a CAM2, you get a CAM7. And so we said, well, it showed up in the pathogen response network. It's really, really, really closely related, even more so than any of the other ones that are here. So we're going to look at it, at the very least, just to kind of say, oh yeah, our network works. It wasn't supposed to be a touch responsive one, and it's not a touch responsive one, or something like that, right? So kept it in there just to get a look at it. So we have, in response to touch, we have our group of touch responsive ones, plus, like I said, CAM7 is in there. It's not touch responsive as far as the literature says, but this is the group of genes. We wanted to see, okay, are they really, after we do our experiments on them, and I did my experiments on them with the help of Gabby and Savannah and a few other people, after we've experimented on them, can we say, yes, they're touch responsive, or no, maybe not? And so one of the typical ways that people especially in plant biology, go about that is to, instead of looking at just wild type, you find plants that have each one of these genes, so individually. So I want a plant that can't make CML49, for instance, or CML24, for instance. And then you see what does the plant do differently. Okay, so you mutate the gene and see how is it different from wild type, and in this case in response to touch. Luckily I didn't have to generate these mutants because in Arabidopsis there are an enormous number of tools out there for both generating and then also acquiring previously generated mutants. So we got all of these from the Arabidopsis Biological Resource Center at Ohio State University. I think, yeah, all of these came from there. And we came and we just had to verify that they were mutants for sure. So check them, make sure they're homozygous, and then start experimenting on them. So like I said, wanted to see, are each of these genes somehow involved in the response to touch by seeing you get rid of that gene, you knock it out, as we say, and see how it's different after touch stimulus. And then secondarily, because we are very much interested in also the pathogen response and maybe how those two interact, we want to see separately how pathogen responses are they. All right, so first, in order to do this, we're going to start with the touch response. And what we did was take each one of our mutants, and this is kind of a painstaking process, but you take each individual and you grow up enough of them, and then you can give them a touch treatment or later on a pathogen treatment, and then see how they respond genetically. So do they increase the production of genes in their gene products? How is that different from the wild type response? And in this case, we have some known, so I don't know if everybody can see this back here, but this is the key. I should move my thing over, but we have known touch responsive genes. I've talked about them all before. Touch 1, 2, 3, and 4. When plant perceives a touch stimulus, these genes go up in expression in wild type. And so we started by looking there. Okay, so give them a touch. What does that touch response look like at the genetic level with the expression of these genes? And so in wild type, everything that you're going to see from, in terms of the genetic response, both here and in the pathogen response, is going to be relative to the wild type, because what we can do with, in this case, it's qPCR, so it's quantitative polymerase chain reaction. We're going to look at the expression of each one of these genes relative to wild type. So Columbia wild type is going to be our baseline set at 1. And then, so we have all of our mutants. So like I said, do each one individually. Look, I take one mutant, CAM2, CAM7 mutant, CMO5, do all of them individually, and look first when you don't do anything to them, you just leave them, let them grow nicely. What is the expression of these touch responsive genes like? So what's the baseline level that we're looking at so that we can compare back to? And in this case, wild type is 1, and so anything below that is going to be a reduction in the expression of these genes, so touch 1 through 4. And above that, and so you'll see that in green. Green is going to be a reduction, and then above that it's going to be red, and so you'll have a relative to wild type an increase in expression. And so from here you can see that quite a few are the same, which is indicated by black. They're not statistically significantly different from wild type, and then the majority of them, just starting out baseline, there's a reduction in the expression of these touch response genes. Important to point out is that the gray, like I said, CMO24 is touch 2, CMO12 is touch 3, CAM2 is touch 1. So the gray indicates that this is a knockout of that gene, so there's no expression there. So, we look at it and we say, okay, there's a reduction overall in all of these mutants. There's pretty interesting reduction in the expression of these genes on the baseline. And then you go to, this is what we did our touching with to begin with. We've also redone this by spraying the plants with water. So this is it's may not be clear what this is. This is an aluminum bar that you can hold in your hand, and it just fits right into a well and a 12-well plate. It's like a cell culture plate. Grow the plants in there, and then you put the bar, rest it with the weight of gravity, no additional pressure for a couple seconds, take it off, and then that's interpreted as a touch stimulus, and look at what's happening. So we're going to touch the plants. So 30 minutes after the touch, in wild type, you have an increase in expression of the touch genes. So with the most significant increase in touch 4, it goes up by 20-fold, which is pretty big. And then we're going to compare the mutants to that. So again, so we touch them. Again, they do, on the whole, each one of the mutants does have an increase, but it's still pretty, or not all of them, but quite a few of the mutants, on the whole, do increase in the expression of touch-related genes after the touch stimulus, but it's way, way down, and if you compare it back to where they started at 0 minute or in the untreated samples, it's not a significant increase for most of them. So where you expect a really large increase because of what wild type does, you don't seem to get that with most of the mutants. And then you do the same thing and you wait 60 minutes and see what the genes are doing. And by this time, wild type has kind of started to come back down, return back down to kind of the unstimulated levels almost, just like, on its way down there. And again, looking at the mutants, they're all pretty far down. And there are a couple of notable exceptions here. There's CML24. In the CML24 mutant, you do have a relatively high expression of CAM2, which is touch 1 here. So that's interesting that maybe there's some connection between touch 1 and touch 3. But overall, pretty steep reduction. So that gave us a good indication that maybe we're on the right track. They aren't doing what wild type does in terms of the molecular response. So maybe we're going in the right direction here. So then what we did was we used this contraption here, which everybody can go see down in, you can all hang out in Simon's office immediately after here and rummage through his things. And right on kind of the antechamber to his office, which every professor gets an antechamber, right? That's normal. You have this growth contraption here, and you can see clearly on the top you have pots to grow plants in, and then on the bottom you have the same thing here just to demonstrate what the rack looks like. We don't have the pots there. So you have 2 levels. Both levels are illuminated. You can see the lights on up here. Thanks to Sarah Friedrich for taking these great photos. It's a functional piece of machinery, so it's a tape everywhere, and there's wiring that's hanging off. But overall, it's a really cool piece of machinery. And what it does, it doesn't just grow. It's not just for growing the plants there. It's and in a well-lit area. You also get this, what we call the touch bar here, or the stimulation rod. I don't know what to call it, but But what you have is a bar that has 2 motors on either end that are driven by belts, and they drag pieces of whatever material you want. In this case, we use polyethylene sheets because they're flexible but kind of stiff. They don't usually destroy the plants. They don't usually uproot them, which is an important problem to think about. And you can set it to stimulate using the interface up here. You can set it to stimulate the plants as frequently as you like, and that's nice. And so, and then, importantly, because this exists other places, this kind of like automated stimulation of the plants that people have this, but we also have connected up on the very top of both levels, you have these little Raspberry Pi computers that are networked with these little cameras that are suspended. You can see here, they're suspended over each one of the flats and are imaging 9 plants at a time. And so what we can do is not just stimulate them and kind of see what happens at the end, but we can watch over time what's happening. And I will give you a look at that. This is pretty interesting. So I'll talk a little bit about this later, but just like watch the video for now. So this is what's happening. This is very sped up, obviously. This is 8 days worth of touching, but they're already a couple of weeks old when we start the touching, so that they don't just die right away, because if they're super duper small, you know, they just get battered. But once they're a little bit older, I hope you guys all saw that, but here I can hopefully do it again. So they're growing fine, and then the touch sheets come and wipe back and forth every 5 minutes. And like I said, over 8 days you get to watch the whole thing. And then developed by one of our good friends and somebody from Edgar Spalding's lab, Nate Miller, developed software that allows us to take the images that are captured and analyze the growth that's happening in these plants. And so what you start out is with this 9x9 stratum, or with all the plants here, or 3x3, 9 plant layout. And then in the top corner, you see here, this is like the idealized version, but you have a QR code that is read by the camera. It tells what plant is there, so like what genotype, which mutant we're talking about, which position the plant is in, so we can keep track of them as we go on, and then any other information that you want can be encoded in there. But so the camera takes a picture of each one of the plants, and then you can measure their responses to touch in terms of the growth rate, or you can look at individual leaves, or you can look at the elongation of the petioles, so the connection from the base of the stem to the leaf itself, the blade of the leaf. And so that gives you a good look. So the software quantifies the number of green pixels here, and then you can say this particular leaf is a leaf, and this one is a different leaf, and kind of compare what's going on there if you like. There's a lot of flexibility in the system. What I'm going to talk about is kind of just really simple but really interesting aspect of the overall growth morphology that's happening. So, or the change in morphology over time. So, I'm just going to quickly break down how we classify the responses to touch. First, class 1, no difference from wild type. All right, Columbia 0, there's just no statistically significant difference from wild type. Class 2 is going to be the untouched plants, so unstimulated, just growing normally, look like wild type, but when you touch them repeatedly like we did, the mutant plants are significantly less sensitive to touch, which will mean that they grow much better, much larger than wild type does when it's touched. And then class 3 is going to be, even when they're not being touched, the plants are dwarfed. It could be some other mechanism, but it might be because when you compare the touched and the untouched plants, they're not significantly different in size from each other. So maybe what's going on is that the plant is always perceiving this touch is kind of always on. That's possible. So I'm going to show you kind of what this looks like. So we have these figures. So you have the growth over time, starting at day 0, hour 0, minute 0, and increasing. So the top line, the dashed line, is going to be, these are untouched, left alone. You monitor their growth, and then like you can see here, they're 8 days, a little over 8 days here. And then the bottom solid line is when they're touched, how large do the rosettes, the entire leaf structure, how big do those get? And you can kind of compare them, and you see that there's a pretty significant reduction in overall growth when plants are routinely, continuously stimulated. So that's pretty interesting. This is just a wild type, so it kind of defines class 1. Class 1 is things that are like wild type. I'm not going to go through all of them because it's a lot, it's 11 mutants and wild type. So I'm just going to pick out a couple of each that are kind of exemplify what each class is like. So class 1, we have, interestingly, we have CAM7. Now it looks to the eye like there's a reduction, but this isn't statistically significant. So CAM7 kind of fits into class 1 as not being touch responsive. Class 2, so grows like wild type when it's untouched, and then there's a really big difference when it's touched. So in case of class 2 and in CAM2, instead of this touch-related reduction, this really large reduction, they're relatively insensitive to touch, relative to wild type there. So that was pretty interesting. And then class 3, so CML12, one of our other touch genes, is here, and you see that there's pretty significant just unstimulated reduction in size, and then they're not that different from wild type when they're touched. And then again, another class 3, kind of like the really extreme example of that is their CML24 is very dwarfed when it's untouched, I mean relative to wild type here, and then there's no significant change even after touching. So what you would expect if this was, you know, not a touch response thing is you would expect, okay, so even though they grow smaller overall when they're untouched, when you touch them, there should be an even further reduction. And it might be that we've hit kind of like the lower physiological potential, or the limit for how much growth you can get over this time, but it might be because of the touch-related phenotype. So, but this is kind of overall what it looks like. So we did this for every single one of the mutants, and you get kind of a graph like this, where you can compare the untouched wild type here, and then you have the touched wild type. You can see there's a pretty significant difference between these, as we saw in the chart before. When you look at CAM2, there's not a difference between these, but there is a significant difference between touched CAM2 and touched wild type. And so you can go through and look at all of them, and what you get are the 2 classes, or the 3 classes like I was talking about before. So you have those that are not different from wild type, so like CAM7 here. And interestingly, CML16 didn't look like it was significantly different either. And then all of our 2s are those that untouched, they grow like wild type, but when you touch them, they're on average significantly larger than wild type is when it's touched. And then the 3s, you have a pretty significant reduction in size, and then really not much of a change after touching. So they look similar whether they're touched or not. So, so that's kind of what we got. So we have, this is one of the results. So that's what we're looking at when it comes to the touch response. So now what we wanted to do is, okay, we've got a pretty good idea of where each one of these genes lies in terms of its involvement in the response to touch. Next, we're going to look at involvement in pathogen stimuli. So, like I said, we have these. This is the network again. CAM7 doesn't seem to be involved in touch. CML16 maybe not be involved in the response to touch, but the rest of them maybe. So, talking about how plants respond to pathogens. First, we have calmodulin over there, like we have seen before. The plant cell is constantly keeping the concentration of calcium on the outside of the cell higher than on the inside. And therefore, when you have a stimulus, let's say when a bacterium comes along, you have what's called an elicitor in this case. So it's something that elicits a response in the cell. In this case, it's flag 22. This elicitor is bound by its pattern recognition receptor, which is a protein that sits on the outside of the cell, looking for these patterns, these elicitors. That leads to a rapid influx of calcium in the cell, which we talked about before, how calcium floods into the cells. And then calmodulin binds those calcium ions, and then it goes on to regulate certain things. So this is kind of like a figure describing what happens when it happens. And one thing you'll see is that calmodulin, and there's a lot of research about this, but calmodulin is involved in the expression of genes that are involved in the production of salicylic acid. And so one of the big things that a plant cell does when it detects a pathogen is it tries to make the cell inhospitable to the bacterium. And it also wants to signal to itself and other cells around it that there's a bacterium there and everybody needs to react. And so salicylic acid is one of the not early, but like kind of early steps in taking those further steps to prevent further pathogen infection. But one of the really early things you see after calcium influx is increase in expression of these genes. So CBP60G will be one that I'm talking about. SR1 will be the one that I talk about. And ICS1 is important because it leads to salicylic acid production and kind of helping the cell respond to the pathogen. So we thought, because calmodulin has been shown to be involved in this and be good candidates, we are going to look at, okay, we have our touch cams and CMLs. We've assayed now, how do they respond to the pathogen? So we did the same thing. We looked at the molecular response here. So like I said, we have our SR1, CBP60G, ICS1. Those are the ones that are involved in salicylic acid that should boost the defenses of the plant. Then we have MPK3 and MPK6 that are involved in other signaling in response to pathogens, but also very important. So we looked at the expression of these in wild type, again, wild type's 1 for being the baseline. And then you see that there's a pretty significant increase, baseline level, unstimulated, in these pathogen response marker genes, and quite a few of our mutants here. Interestingly to me, and I wish I had time to talk about this today, but CAM2 and CAM7 have pretty high levels all the time, and that's cool. But we're going to treat them with our pathogen elicitor, so we're going to take FLAG22, treat the plants, and look at what happens. And so in wild type, you get a pretty massive increase in CBP60G and MPK3 expression. And then you compare that with the mutants. And CAM2 and CAM7 don't have a significant increase relative to their untreated. There's not a really big increase, but it is down relative to wild type. Some of these you get a pretty big jump in pathogen response marker genes after 30 minutes of treatment. Overall, things are pretty high relative to wild type, except for MPK3, which is interesting. One of our other signaling pathway genes is down relative to wild type in most of these mutants. And then after 60 minutes, you look at it again. Wild type is starting to come down, and you look at the mutants, and the pattern kind of continues. So we had an idea that maybe we're right, maybe a lot of these are pathogen responses based on the molecular response. We might be getting into too much detail, so I'm going to keep moving. But so we looked at the physiological response. We used the pathogen that this elicitor is from, this FLAG22 elicitor. We're going to use Pseudomonas syringae. We're going to use a relatively high concentration of that. And first, we're going to look how many days relative to wild type do they survive. So again, we're going to go through and see what our classes are going to be. Class 1 is going to be the pathogen response is not significantly different from wild type. 2 is going to be the plants, when you treat them with the bacterium, they survive significantly more days than wild type, and fewer bacteria grow inside the plants. And I'll show you both of those. And then class 3 will be they survive significantly fewer days than wild type, and more bacteria grow inside the plants. And so I kind of have that. First, we looked at how their survival is. Here you can see the ones on the left next to wild type. Wild type usually survive around 6 days after pathogen infection. These survive significantly longer, 2, 7, 5, 9. 12, 16, 24, and 40 survive significantly fewer days. And then the other ones were not different from wild type. So we could try to classify them based just upon that and say, okay, the ones that survive significantly longer were 2s, fewer days, 3, and then everybody else is a 1. But we also looked at kind of the proliferation of the bacteria inside the plant. And so we quantified how many bacteria grew just inside of the plant, made it in after infection. So we flooded the plants with bacteria and then counted how many bacteria were on the inside. only. And what you get after 1 day of infection, we have significantly fewer bacteria growing inside of CAM2, CAM7, and CMO5. CMO9 looks like wild type. Most of the rest of them are either higher or they look like wild type. But after 3 days, you have a significant reduction. This is about a 10-fold reduction in the number of bacteria. And we're talking about a difference between 290,000 colony-forming units and 27,000 colony-forming units. So that's, you grind up all the tissue, you plate it on a Petri plate, and then you wait and you count how many colonies start to form. And then you make that relative to the weight, the starting weight of the plant itself. And so what you have here is after 3 days, significant reduction in the number of bacteria relative to wild type, growing inside the plant in 2, 7, 5, and 9, which kind of fits with our previous observation that they will survive longer. And then you have an increase in CMO12, CMO24, and CMO40. And CMO16 looks like wild type. So we go back to our previous slide, and we're going to give CMO16 a 1, because even though it looked like it survived fewer days than wild type, it had about the same number of bacteria growing inside it. So it kind of could be either. So we have that. We have our results from the touch. We're really interested in those that seem to be involved in both. And so if I take away the ones that don't really look like they're involved in both of these, so any of them that have a 1 next to them, they're like wild type, so they might not be involved in that pathway, because if they're like wild type, then even if you knock out that gene, nothing happened different from wild type with respect to how it responded to these things. So not as interested in those. So we have our list of genes here that seem to be involved in both. We're going to fit them into one of these Venn diagrams, which everybody loves, except for our former governor, who did not have any idea how to use one. And so we have the ones that are involved in both. We can say pretty solidly, based on our experiments, that CAM7 doesn't fit in the touch-responsive ones. Our network agrees with that, or our network gave us the hypothesis that it probably isn't involved in the touch response, so that's good. CMO27, CMO44, 46, and 49 all seem to be touch responsive, but not involved in. They're involved in the touch response, but maybe not in the pathogen response pathway. CMO16 seems to fit there too, although based on how it responded or didn't have a differential response to touch, maybe it doesn't fit in either. We don't know. And so that's basically it. So we looked at all of these things, and we had suspicions, and we checked them out and found out that maybe not all of them are involved in both responses, but we have a pretty good idea of those that could be, and they are fruits for future research. And with that, I want to thank everybody in the Gilroy lab, current and previous. Thanks for those of you that were able to be here. Especially want to thank Gabby and Savannah, lovely, very, very helpful people, and I wouldn't have been able to do what I did without them. I wish I could have shown you like everything else that we got done, but you just have to read the dissertation. That's so everybody else in the lab, Nate Miller, I gotta point out, even though he's not here because he's hanging out with his family. He helped a lot. Makoto, thanks for being here. Thanks everybody. That's it. Traditionally, we leave a little bit of time at the end for questions. Does anybody have any questions for me? Please say no. No, I'm just kidding. Okay. Dick.
So I'll just let you speculate, but for the mutants that showed elevated resistance to Pseudomonas,
Yeah.
would you suspect that they have any phenotypes related to stomatal aperture
Yeah. Yeah, I mean, so there is evidence that some of the CMLs, a couple of the CMLs might be involved in regulating the opening and closing of the stomata, the holes in the leaves that are a potential avenue for bacteria to get in. Yeah, I mean, I think that would be a good thing to look at. There are definitely tools out there, microscopy tools that people can use to look at those things. And you can even artificially induce different either opening or closing of the stomata with hormones and things like that, and kind of do that in wild type and compare it to the mutants. I think that's a good idea. Yeah, sure could be. Yeah.
So the touch mutations are pretty cute, but one thing I was wondering about, how can you rule out that you just have sick plants due to mutations that aren't the target mutations that were provided to you?
Yeah, I mean, one of the ways that you can kind of try to do that is look at them overall physiologically and just say, do they grow healthily, even if they're smaller?
Well, the classic is to do some sort of alpha mutation test.
Yeah, absolutely. So a lot of times what people will do, try to reconstitute the wild type using complementation. Doing a lot of back crosses. I mean, yeah, that's a good point. They could be just sick plants, but when we look at them, I mean, other than they seem a little smaller than wild type when they're that young. When they grow full, they produce seed, they reproduce well, they seem very, very healthy, but when they're about 2 to 3 weeks old, right, they're dwarfed, but might just be the developmental stage. And that also could be masking some of the other touch-related phenotypes that we would otherwise see. But that's a good point. Yeah. Thanks. Anybody else? Okay.
All right.
All right.
I guess that concludes the public
Yeah.
process.
Grab donuts and coffee on the way out, please. And Johnny made a delicious pineapple upside down cake that people should eat as well. And donuts are from Greenbush, and everybody loves Greenbush, so enjoy. Otherwise, they're going in the office, and you don't want those schmucks to be there.